Thank you. So good morning, everybody. So as soon as everyone is seated, I will talk about information processing in biological systems, the integration of information. And this morning we'll take a very molecular perspective on this aspect, and in particular a very synthetic perspective. We'll try to build models of the ways cells create information processing systems. So do not hesitate to interrupt during the presentation and ask questions if, or remarks if everything is unclear. I prefer to have the presentation interactive rather than talking alone for 1 hour and a half. Okay, so we are all used to the fact that living organisms process information, they compute. And we also know that living organisms are basically molecular systems, right? So if you take this animal in particular, it can think, it can react to the environmental clues, and so on and so on. But basically, we assume that these information processing capabilities come from the fact that the animal has a brain, a nervous system. But it happens that we have also discovered information processing within biological systems at a smaller scale. And this is an example of, for example, a single cell. This is a white blood cell that you are looking here. This is a big thing in the middle. And the small black yo-yo that you see here is a bacteria. And currently, the white blood cell is doing its job, is chasing after the bacteria, and it just got it now, and it will digest it in order to protect the organisms. So when we look at this movie, we also have a strong sense of precision and precise information processing, precise chasing after the bacteria, and of course, the scale is much lower than the full chick, and there is no brain, there is no small computer inside this relatively simple bag of chemicals. This was still a eukaryotic cell, a relatively complex cell, but you can go down in complexity, take a bacteria, even take a virus, and you will also find ways of and information processing capabilities to some extent. For example, I don't know for example, in the case of this simple virus, a genetic switch was discovered and studied at length. So how do they do that? Well, inside, as I just mentioned, inside of these systems, it's relatively simple. It's mostly a bag of relatively amorphous soup. For more complex cells, you have a little bit of organization, but it's not going to be the point here today. So we will take these systems as a bag of chemical and ask the question of how these bags of chemical perform relatively complex and, of course, useful for them computations. So, the question is, where is the computer? So this question was the question was started to be answered in the 60s, for example, with this historical example of the operon lactose by a French scientist, Jacob and Monod. And the system they were studying was a relatively simple and classic bacteria, E. coli. And it was known that these bacteria can live on different types of food, for example, glucose and galactose. Okay, so both sugars allow the bacteria to grow and to divide ultimately. But the bacteria's preferences is prefer glucose because it's more simple to digest. So there is a direct. It's very simple to relatively simple to integrate into its metabolism. Galactose is a bit more complex. It requires a pre-processing step done by an enzyme called beta galactosidase. Right. So basically, what's the purpose, the control function that the bacteria needs to operate is something like that. So when the preferred food, glucose, is present, whether or not galactose is there, it is not necessary to activate the production of this particular enzyme. That would be wasteful because in any case, glucose is present, right? So this pathway should be activated only if there is no glucose, but there is galactose, right? If there is no glucose and no galactose, other things more complex may happen, or the bacteria may die if there is no food at all. But we have these three cases, and then the control problem is relatively simple. You have this table, you need to fulfill it. So how does this happen? Well, there's some small problems with the PowerPoint extraction of the presentation, but yes, okay, this one is going to be more clear. So we can represent this problem as a small network. We have a genetic element that's going to encode the genes for the enzymes that allow the processing of lactose. So if these are expressed, then lactose processing is enabled. And in order not to express this pathway when not necessary, there is a repressor. So this is basically repressed, right? And you also have an activator. So now there is link. The system creates link between glucose, so the two inputs, glucose and lactose of the system. So glucose and lactose. And the connection with this simple network of gene encoding, repressor, and activation is such that you fulfill this particular table. So you can look at if there is glucose, for example, the activator is repressed, and then whether or not you have lactose, you don't activate the system because this is repressed. There is no activation, right? On the other hand, if there is no glucose, this guy gets activated, and the repressor, and in the presence of lactose, the repressor is repressed. So this is a double negative, comes down to an activation, and the system lights on. Okay. So what we are observing here is a relatively simple network, right? You just have a couple of nodes, a couple of links that performs a useful control function for a living organism, right? And so this was more or less the initial discovery of this concept of genetic regulation and chemical regulation inside of cells. But over the years, many more examples were discovered, many more molecular ways to perform computations that are useful for an organism fitness. And this is another classical example. Here we have, again, a bacteria. It's growing, so integrating more and more carbon and producing new molecules. So that's what we see here. So starting from a small bacteria, we come up to a large bacteria, and now it's time to divide into two bacteria, right? And before the bacteria divides, it needs to decide where to put the boundary between the two new cells, right? So we have to cut into two parts. And the best way to cut into two parts is to create these two parts perfectly equal. Right? That will create two daughter cells with optimal fitnesses, maximum chances of surviving if both of them have the same size. If one is too small, then it's probably not gonna survive, and it's just a wasteful division event. Okay? So the question is, if you are a bacteria, again, relatively simple bag of chemical, how do you perform the computation that comes down to finding the middle of a segment, right? So this question was investigated for some time, and the discovery was again a small molecular network, which is called the mean system, and whose function is exactly that. The idea is that the implementation of reaction between these three components, here we have three proteins, is going to allow the detection of the middle of a segment. So how does it work? So we will have a small first idea here. In this case, one of the protein of the system. So we have three proteins here: mean C, mean D, and mean E. So one of them has been tagged with a fluorescent reporter, so that we can observe its localization. And here we are looking at the microscopic image of a bacteria. So you see the segment here in bright field. And this is time. So as time flies, you see that this particular protein complex with the mean protein and the fluorescent reporter change location from one pole to the other one. And this basically creates a wave that goes back and forth, bouncing against each pole, right? So this is what one of the enzyme protein does. And we have a small cartoon here. In fact, the other enzymes are also associated. So the mechanism by which this is obtained is that enzymes associate to the membrane, and then they will change the binding constant to the membrane dynamically upon the reaction with the other partner in the network. And the end product, we will not go into the detail, but the end result is that you have this wave of oscillations where one of the enzymes here covers the pole, and then it starts to detach. This reaction accelerates, and they reattach on the other side. And then the second enzyme comes, triggers the reaction, so everybody detach and goes on the other side, and so on and so on. And you see here a cartoon representation of the enzyme concentration across time and space. Space being the long axis of the bacteria. So one, as you see, the green one, the one that was tagged beforehand, oscillates from pole to poles, and the other one tries to follow. And you have a third one that, by trying to follow this oscillation, in the end, if you integrate its concentration over time, you precisely discover the middle of the bacteria. And so downstream of this system, there's a cascade of new events that's going to trigger the creation of the Z ring, and after some more molecular events, it's going to close the bacteria and create two new daughter cells. Okay. Oops. So we'll not go into the details of how this network works, but the message is that over the years, many more systems have been discovered, and now we know that some of them are very complex, and the most complex of them, of course, they control more complex tasks. For example, this is a representation of what we knew in 2006, at least, about the regulatory network that controls the life cycle of budding yeast. And Okay. So and it's the same for this white blood cell that we were observing in the movie at the beginning. It's also a network of chemical events that allows it to very precisely track and grab a bacteria, which is in a way relatively impressive. Okay, so these molecular networks are fundamental building blocks of biological system. In a sense, they are inevitable because the very essence of a living system is to reproduce. So to implement a network that starts from itself to create itself, right? A relatively direct feedback loop. So starting from this network, they need to be there just because of this very process of replication. And life is using them to perform more complex functions and in particular computation. So what about molecular networks? Are they special compared to other networks, such as, I don't know, neural networks, mechanical networks, electronic networks? Do they have something very special? So I like to connect this question to a very similar question that was asked maybe 200 years ago when people discovered organic molecules. And so chemists were used to inorganic molecules, and they were already manipulating them and making reactions with them. And then they discovered the organic molecules as constituent of living systems. And the question at the time was, are these organic molecules very special? Are they different? Is it because they are organic that they are living, or is it just because something else? And so the answer to this question, in the end, came up with the creation of synthetic chemistry, right? Synthetic chemistry allows the synthesis of those first, or the first simple at the beginning, and then more and more complex organic molecules. And people discovered that they were not particularly special. It was possible to synthesize them outside of living organisms. So they were not the essence of life, right? They were just a different type of molecule that people had not thought about. And so it's a bit similar now for molecular networks. Are they special, or are we just unused to manipulating them? And maybe to answer this question, and a part of my research is to say that maybe to answer this question, we need to try and go and build more and more molecular networks as models of cells, and maybe also for possible applications. So if we try to answer the question, it's a kind of a mixed answer. We can say no, we can say yes, and we can say, but yeah, basically we don't know. So the no answer is because we know that networks basically share some fundamental properties. We will discuss a bit more about what are those fundamental properties that are shared across all networks, whatever they are instantiated with. And then we can also say yes, because we have a network that performs, for example, computation in cellular networks. But the edge of those networks are something very different than an electronic component. They are molecules. So the nodes are molecules, and the edge are reactions or interactions between molecules. So that's relatively unusual and quite specific. And then you can also say we don't really know, because we don't have much experience about building molecules, this kind of molecular networks. Most of the one we know they come from biology. They are the ones that I introduced just before, for example. So maybe to better answer this question, we will need to try to build more of them. So about universality of networks. So here we have a representative of a network, and one of the most fundamental universal feature in network theory is that the higher level properties of this kind of network, they emerge largely from the topological structure of the network, and not from the real identity of the node. So you can represent this network with A, B, and C, or you can represent it without saying what are A, B, and C. And basically, a large part of the property of that particular network will remain whoever is A, whoever is B, and whatever is C. And to exemplify this fact, I selected this relatively also classic example of the Lotka-Volterra equations, or it is this particular set of differential equations that you see here, which basically represent this network with two components, the X and the Y that you find here. And in this particular network, so X self-activates, and Y also self-activates, but with consumption of X, right? And then you have a decay pathway for Y. Okay, so those are the classic Lotka-Volterra equations. So you find the same, the idea of this, the mathematical description of the dynamic of this network here. X self-activates, so the rate of X production is proportional to itself, the positive term here, and Y also self-activates, so the rate again is proportional to Y and X because it needs X. And this term you find because X is consumed, so it disappears from here, and then you have a simple first order decay, right? And so it happens that in fact these Lotka-Volterra equations were originally derived to discuss chemical systems or animal populations in particular, such as this particular network, which is called the predator-prey network. So here X is a prey, for example, rabbits, and Y are predators, for example, fox. And relatively naturally, we understand that indeed the prey have the ability to reproduce themselves, exactly same as the predators. They also reproduce themselves, but before doing that, they want to have a small lunch of prey. So they will consume a prey, and then maybe they can replicate. And this system is famous for one reason: is that relatively simple network with just two components, but it still shows very interesting dynamic, and in particular oscillatory dynamics. It is known that if you solve this equation, you will find oscillatory solutions, limit cycles. And indeed, for animal population, and in part, this is an example of two animals, a rabbit and not a fox, but a lynx. So it's a pair of a prey and a predator that is relatively isolated from the rest of the ecosystem, right? So by this I mean that the lynx mostly feeds on those rabbits, and the rabbits, their main predator are lynx. So it's relatively clean implementation of this simple mathematical system. And indeed, it was observed that if you look at the population of those animals, which is here measured by the amount that can be captured by hunters, you find that you have strongly coupled, strong oscillations over time. So these are years, and these are the catches of the two animals. And over time, you see strong oscillations in the populations, and they are coupled. So apparently, it's indeed this particular rabbit and this particular lynx in this particular ecosystem, they do implement clean Lotka-Volterra oscillatory dynamic. But you don't need to. I mean, you can replace rabbit and lynx by completely different things. So here, the same Lotka-Volterra dynamics of the same network can be used to discuss a problem that I'm much less familiar with, but dust-forming plasmas. Apparently, you have some types of stellar entities that play the role of the rabbit and others that play the role of the fox, and altogether they implement a very similar dynamic. And this is one example, but you can find other examples in the the same field, so the same kind of network and the same kind of dynamic can explain completely different things. So by this, I just want to exemplify that you can replace the identity of the node. You will maintain the most important part of the global dynamic and function, right? So, and in the particular case of this system, in fact, Alfred Lotka discussed this set of equations to try to propose a model explaining the first observations of oscillatory systems in chemistry. So at the time, it was something like 120 years ago, at the beginning of the 20th century. So people were observing oscillatory behavior in chemical reactions, and they were very surprised because thermodynamics, if you interpret it relatively trivially, seems to forbid this kind of behavior. So you should go to equilibrium and not oscillate over time. And so people were very surprised. They were discussing, is it possible, is it impossible? And Lotka said, well, we can at least write a small kinetic model whose solutions are indeed oscillatory solutions. And so he suggested this particular system. And Volterra, on his side, was discussing the same system in the field of ecosystems. So in principle, it would indeed be possible to define the molecular prey, molecular prey and a molecular predator, put this in a molecular soup, and if the network is well implemented, it should be possible to observe oscillations of those two chemicals and then to create a clock or to perform some kind of time processing function. But it happened that such a system was never discovered over something like 100 of years. So we will come back later to the first implementation of molecular predator-prey oscillator. Okay, so let's go back to our cells and their information processing network. So these exist as various flavors. In fact, you have various types that have been classified. For example, gene regulatory networks and signaling cascades, enzymatic networks, and metabolic networks. So they are all relatively different in terms of the chemistry that they use, right? And also somehow the way they work. So today we will mostly talk about gene regulatory networks because they have some very they are very important, obviously, for cells, and they have some nice characteristics when in the purpose of building other networks. This is an example of a gene regulatory network. I forgot exactly what this guy is doing, but I just wanted to give you the idea that they can be very complex. They can become very complex. They can include many nodes, many interactions, but conceptually they remain relatively simple. And the reason for that is that they have a very modular architecture. And you find always the same motif, or more or less. I am simplifying grossly, but more or less you find the same motif over and over. And the large network that you have just seen on the previous slide is just a repetition of this basic motif again and again in between many nodes. And so the motif goes as follows. So you start from a gene. And as you know, gene, they encode proteins. And you go to the gene, from the gene to the protein by two stages, which are chemically very complex, but they can be represented as a simple chemical arrows here. So the first one is called transcription. So the information encoded into the DNA is converted, is written again in a messenger RNA molecule. Right? So you go from DNA to RNA, and then you have the second stage, which is called translation, where the same information is interpreted now in another polymer, a polypeptide, protein, the process of translation, right? And this is how basically your genome encodes for protein, which are the phenotypic elements of cells. So many of those genes, they encode for proteins that are useful from a physical-chemical point of view. They can support your membrane, they can convert food into energy, such as the beta-galactosidase that we have discussed before. They can do many different tasks that are useful on a physical-chemical point of view, right? But some of them, they don't do anything like that. They don't have catalytic function. They don't have structural function. The only function is to regulate, the expression of another gene. Right? So we express a gene, we express a protein from a gene just to affect the expression of another gene into another protein. Right? And this regulation typically goes by having this protein here bind close to the position on the genome where this protein is encoded. So it binds there, and by binding, it can affect the process of transcription, for example. Okay, it's relatively easy to understand that maybe binding on the gene or very close to the start of the gene will inhibit the transcription of, because it will limit the access of the gene to the machine that is required for transcription, the polymerase. Okay, so this would be a negative regulation, but you also have many examples of positive regulations, similar. So what is interesting in this particular small block of chemical element is that you start from a gene. And the output is also the regulation of another gene. So it means that if this gene does the same thing, express another protein that affects gene 3, then you can start to create a cascade. And by connecting many of these modules, because they always start from a gene and go to a gene, you can connect them and create the large network that I was discussing before. So this motif, this conceptually simple motif, chemically very complex but conceptually simple motif, can be cascaded over and over to create large networks. And this was very well, I need to say something beforehand. So what are the key elements of these regulatory networks made of the cascading of many of these small motifs? So as we mentioned, the topology of this network is going to be determinant to the function. But of course, it's not only the topology. You need to take care of the kinetics, the kinetic laws and the kinetic rates. The laws because they affect the linearity and non-linearity of the element in the mathematical description of the system, and so they affect the dynamics. And the rates because they will position your system in parameter space. And you know that for those dynamical systems, you can have very different behaviors depending on where exactly you are in parameter space. This is something we will exemplify later on. And in the case of spatially distributed systems, you also need to take into account the diffusion rates and the boundary condition, but this is not something we're going to discuss much today. The item that we don't have here is the chemical, right? So we don't really care about the chemicals. No, there is no direct link between the physical-chemical nature of the parts and the function. And this is the point where I wanted to go. This was very well exemplified by the first papers of a field called synthetic biology. And here is an example from 2000 by Elovitz and Leibler. So it was the beginning of the first synthetic biology, so in vivo, artificial in vivo oscillatory networks. So their goal was to demonstrate that you can use the cell as a kind of platform, as a kind of machine, put your own artificial network inside of the cell, and have the cell implement the dynamic you wanted to encode in this particular network. And so in this case, the goal was to implement oscillatory behavior. And the network they decided to perform this function is this one. So it's a network with three nodes linked by 3 negative interactions, so 3 repetitions in a loop. And so you can indeed imagine that that such a system is capable of oscillations. So imagine you start from a high concentration of this particular component, right? So this particular component is high, and the 2 other guys are low. So this one is gonna, because it's high, it's gonna replace the expression of this one. It's not gonna increase. But because this one is low, then this one is not repressed anymore, right? The link is not very active. So this one can go high, it grows. And as it increases, it's now repressing expression of this one. So this one, the concentration of this one is going to decrease. Because this one is decreasing, this one is not repressed much anymore. So it's going to increase. Because it increases, this one is going to decrease, and so on and so on. And you go in a loop, like a kind of backward loop, where they increase sequentially and repress the next one and be killed by the one that is behind them, because they repressed the one that was killing the guy that repressed them. So the important point here is that to implement this particular network, and hence this particular function, the researchers used parts that have nothing to do with any oscillator in any kind of organism. So they selected 3 genes. Theta is a regulator which is involved in antibiotic resistance in bacteria, this one. This one is a metabolic regulator from E. coli that controls the expression of sugar processing enzymes, so nothing to do with cell cycle or oscillatory behavior. And the third one comes from a virus, a phage, and it controls the switching between 2 lifestyles of this particular virus, lytic and isogenic. Okay, so these parts have nothing to do with the end function. Just by connecting this part into the proper architecture, then indeed you can obtain the desired function, which was represented here. So here we are following a fluorescent signal over time for single bacteria. We don't see where the fluorescence comes from here on this schematic, but there is a fluorescent reporter that allows to follow the concentration of those proteins. And indeed, you see that the system started to oscillate and implement the desired dynamics. Okay. If you have questions, I really do not hesitate to interrupt and ask any question. So far, it is an artificial. This is completely artificial network, nothing to do with any natural function, but it is still implemented inside of a living organism. So if we really want to model those systems, maybe we should also try to simplify the model and get rid of the living organism. So one way to do that is to just kill. So this is inside of the bacteria. So maybe you can just kill the bacteria, takes the internal content of the bacteria, and put your new network inside of this chemical soup. And if you are lucky enough, maybe you can have similar types of behavior outside of a living organism, using the same machinery, the same expression platform, but completely outside of the living organism. So this was implemented, for example, in this paper by the group of Sebastian Merkel in Switzerland. So again, the goal was to design oscillatory dynamics using parts that have nothing to do with any oscillators in any living organism. So the network they selected is slightly different than the previous one, but if you look at the link, you can probably convince yourself that here you have positive feedback, and then this guy is going to activate this guy, which is going to kill itself. So it's both a positive and a negative feedback, and this is consistent with oscillatory dynamics. And so this system was not introduced inside of a living organism. It was just dispersed into cell juice, bacterial juice, itself being put into microfluidic devices, but this is not really the point. And it was indeed possible to see that concentration of the parts inside of this this chemical soup started to oscillate, as you can see here. So it's having relatively nice oscillations, right? Outside of the living organism, it was a model of what can happen in a real cell. So what we are going to discuss today is one step further, trying to simplify even more, get rid of the cellular juice, use very simplified, I mean, as simplified as possible component, and try to construct indeed completely in vitro now reaction networks with similar characteristics and similar dynamical and information processing functions. And the motivation to do that, as I mentioned, is that they can be taken as model of cellular networks, and maybe we'll learn something about the cellular networks by trying to reproduce them in a completely different context. And maybe there is also a possibility of designing a chemistry of not just processing physical-chemical states, but maybe also processing information. And to do that, we will use synthetic DNA and purified enzymes still. So I will be very brief on this one, but you probably know the structure of DNA. So DNA is a polymer, informational polymer, because it's made out of a number of different building blocks, so 4 in that particular case. And it has a very specific properties of having a tendency to associate 2 polymer chains into one structure, which is helical, which is called the DNA double helix. And this is done with sequence specificity, because the 4 different bases of DNA has a preference for the one with which they like to pair. So adenine likes to go with thymine, and guanine likes to go with cytosine, right? So if you have two pieces of DNA, they will want to associate into this duplex, but they will do that only if you have the correct matching between the sequence of one strand and the sequence of the other strand. And this correct matching goes with a very simple rule that in front of A you want T, and in front of G you want C. Right? So the rule is basic, simple, and easy to understand. So this is very useful because it solves a problem that is typically very difficult to address in chemical system, which is the problem of designing specific binders. So if I give you one random chemical A, it's going to be very difficult to find a chemical B that has a specific interaction strength with A. This is an unsolved problem in chemistry. But if A is a piece of DNA, then this problem becomes trivial because you can find B as being the complementary partner of A. And by adjusting, by adjusting the sequence of B, you can even decide how strong they will interact with each other. For example, you can select B to be shorter or longer, depending on the strength that you want, or you can include a small number of mismatches into B. So you can decide of the time. You can easily decide the thermodynamics or implement the desired thermodynamics of your system. So that's a very interesting property of DNA molecules. And another one is that you don't need to synthesize those guys. You can just
You can just order them over internet, and you will have whatever the sequence you desire. You will be able to get it typically the next day or in a few days. So that's very practical when you want to design many of these binding pairs, which is going to be the task of creating an artificial network. And additionally, you have a number of enzymes. So they come from biology, but they can be used completely outside of biology. And these enzymes perform very useful tasks on DNA. For example, you have the class of enzymes that are called polymerases, whose function is to copy the information encoded into DNA. So it takes one string of DNA and make a copy of that string of DNA. But you have other functions, such as cut and paste, for example, with restriction enzymes and ligases. So they can cut pieces of DNA, they can bring pieces of DNA together and paste them again. You have also nucleases that's going to chop a piece of DNA, and you will completely erase the information it contains. So that's a kind of a delete. And you already see that these operations, they look a bit like the one that you would have in your computer program, for example. And also a practical point, it's not only very easy to obtain a piece of DNA with a given sequence, it's also they're cheap and getting cheaper and cheaper. And it is exemplified by this diagram of where you see over the years the cost of synthesizing DNA. Those are the yellow and red lines, and the cost of reading DNA. And this is a log scale, so they are really strongly decreasing costs. Right? And in fact, these costs, they decrease so fast and so strong that completely new applications of synthetic DNA have recently emerged. And for example, a couple of years ago, DNA was used to store massive amounts of information. Here it was relatively massive amount of information. So here it was a block of data of 200 megabytes. So this is a snapshot of the video that was, this block of data contained in particular a video. This is a snapshot of that video. It's a movie clip. And so this movie clip in its binary representation was converted to DNA encoding, so quaternary encoding. This particular sequence of DNA was synthesized chemically. It was stored at the bottom of a tube. It was very compact, very small, very lightweight. And then the same DNA was injected again in a machine that we call the sequencing machine that allows you to read the sequence of DNA. So it was the quaternary encoding of the video was extracted again, and it was possible to read that video. So it was maybe the first relatively large-scale demonstration of using DNA as a way just to store whatever information you want to store. So kind of DNA hard drive. So this is not going to be the point that I want to discuss today, but just to give you an idea of the fact that these huge price drops and technological breakthroughs that are going on for DNA allow completely new applications. The application that we will more discuss today is the building of artificial dynamical networks. Okay, so I think I have the picture of the video in DNA format that you can see at the bottom of this. The pinkish stuff you see is the video, which is some kind of fun. Okay, so about molecular networks. So many people have, a number of groups have thought about developing ways to create artificial molecular networks. And the idea is that we get inspiration from what happens in a cell. In particular, we want to keep the modularity that I've just explained. so that we have a simple building block that we can connect to other again and again to create whatever networks we want. But of course, we want to simplify the chemistry as much as possible, because the chemistry of protein expression from a gene is extremely complex, and you would like to have a more simple system to do that. So these are two examples of toolboxes that have been proposed for these particular purposes, and in these two cases, the networks that were built are chemical oscillators again. So this is the gene-led systems described by Kim and Winfrey. It simplified the system by getting rid of any proteins. So it's just DNA and RNA. And regulation is directly performed by RNA elements, right? And so it allows you to create networks with particular function. And this one is what we are doing in my group. So it's called the PEN toolbox. So it's a toolbox also for reaction networking. It's even more simple compared to this one, because it also gets rid of RNA. So it's a DNA-only system that allows you to implement various networks, and this is an example of an oscillatory behavior. So how does this work? So I will discuss mostly about this approach here on the right, but just remember that there are some other possibilities, and maybe people will come up with new ideas again. So how does it work? Well, the idea, as you probably remember, is to have a way that some chemicals activate the production of others, right? While keeping the input and the output to be chemically similar. So for gene regulation, input and output were genes. So here we want input and output to be chemically similar, but the input to regulate the activation of the output. So here we're going to go a little bit into basic biochemistry and DNA biochemistry. So I mentioned previously the existence of polymerase, enzymes that can copy DNA. Most of those polymerase, before they copy DNA, they request two things. So the template to be copied, and a trigger to start the process of copying. So, and it's only when the trigger, so a small piece of DNA, properly binds onto the template, that the polymerase comes in the game and extends the trigger with a copy of the template, right? And so by this process, we make a copy of the second part, the right part of the template. And then we have a second enzyme, which in this case is called the nicking enzyme, and it's one of the enzymes that has the property to cut pieces of DNA. And this one does not cut the two strands, it cuts only on one side. And the system is designed so that the nicking enzyme comes in and cuts right between the trigger, so the input, and the part that has just been copied. And so if you look at this process, you start from a small piece of DNA, A, and you have activated the production, you have triggered the production of another small part of DNA, which is called B. And A and B, they don't have the same sequence, but they are chemically very similar. They have single-stranded DNA of a given length, right? So you can probably use B to activate the production of C by a second system and so on. So the way this is encoded, who is activating who, is by the design of the template. If we want A to activate B, then we create a template where the input, the left part here, is complementary to A, and the output, the right part, is complementary to B. Right? So just the physical connection between these two strands that tells the system, okay, now there is a link between A and B, and this link is an activation. Right? So of course, if everybody activates everybody, it's not going to be super interesting, because the system will just grow and grow and grow. So we need a couple more components. We need some links to block, to repress the expression of others. And this is what we perform here. So we have a way of doing inhibition, where this particular strand here can come, bind on the template, and it is a competition between the activator C and this blocking strand. So this strand is an blocks, prevents C from binding, and then it represses the activation of this particular template and the implementation of this particular rule. And to maintain the system out of equilibrium, we also need a way to degrade. If we want oscillation, at some point we need to create stuff, but at another point we need to remove stuff, right? Because we want to go back to the initial state. So if we work in a closed system, we need a way of a chemical sink. And this is degradation here, where we have the enzyme called the exonuclease, which comes and chops these guys into very small pieces. So they stay in the system, of course, but they don't have any more activity. Okay, so we have the three components that we needed: the way to activate, the way to inhibit, and the way to maintain the system out of equilibrium and on a steady state. So from that, we can start to build small networks, which is represented here. So, we initiate from strand alpha. It can come and hybridize on the first template, first rule, and create an output, and then this output can go and bind on another template and create a secondary output, and so on and so on. So for example, here we have a simple activation. So if we inject beta at some point, we inject beta here, so we'll have a production of gamma, but remember that there is degradation of both beta and gamma, so the production rate of gamma is first going to decrease, and then there is no more beta, so you are not producing gamma anymore, and then it also degraded, so it comes back to the initial. So you just get a very simple behavior, which is a spike of gamma by injecting beta in this system. So here we have a similar system, but it loops on itself. So alpha activates its own production, which is implemented by this kind of so-called autocatalytic template or rule. So both input and output are the same sequence. In the presence of alpha, it comes, binds on the input part, and produces the output, which is itself, and then the system self-activates. So this is this self-activation creates an exponential dynamic, but in the presence of a chemical sink, again, at some point, production rate and degradation rate, they will equilibrate, and you reach a steady state. Okay, and this is a kind of the reverse of the first one, where you have some molecule at a steady state, maybe because of this simple autocatalytic loop. And if you inject the inhibitor, you will block replication, so you will go down, but then this guy gets degraded, and then you come back to the initial steady state. Okay, those are for simple networks. So now let's try to make a slightly larger one. So just remember that there is a flow of energy through the systems, and the flow of energy is managed by enzymes, and comes from activated molecules, which are the DNA building blocks. There are triphosphates called DNTPs, so activated DNA building monomers, which are incorporated into DNA strands. So by the polymerase, it uses DNTP and creates new strands of DNA. And then these new strands of DNA, they are cut first by nicking endonuclease, and then by exonuclease. And what you release is the NMP monophosphate, so deactivated monomers, right? So this is the flow of energy, where the energy comes from, and this explains that you can indeed observe non-trivial dynamic. You are in fact completely constantly out of equilibrium. Okay, so to synthesize something, we will first target a behavior. For example, this is maybe a bistable system with two attracting fixed points. Decide for a network topology that is consistent with the behavior that we want. Convert this topology into a circuit or reaction network, chemical reaction networks. Right? And then convert this particular circuit into the sequence of the encoding templates. So the templates that will say, yes, we have a link that says alpha should replicate. Yes, we have a link that says alpha should, alpha or A should kill V, and so on and so on. So once we have designed those templates and the corresponding sequences, we just order them, put them in the soup together with the necessary machinery, the enzymes and the DNTPs, the fuel. And we perform experiments where we look at the dynamic using fluorescent signals. And we can check if we indeed observe the proper dynamical behavior and compare with model predictions, for example. So we were discussing about oscillators so far, so let's keep an oscillating system. So this would be a relaxation oscillator. We already discussed this particular topology. So you have a self-activation process with a short loop, right? And then you have a self-inhibition process with an intermediate, so a long loop, which basically implements a delay. And because of this delay, so the system is nonlinear enough that you can expect it never to reach a steady state and be trapped on the cycle. So to implement this particular topology, we need a number of encoding DNA templates. So we want self-activation for alpha, this is template A, with alpha as input and alpha as output. We want alpha activating the production of the intermediate beta, which is here, so alpha as input and beta as output. And we want intermediate beta to produce an inhibitory compound for the first template here, so beta as input and the inhibitor, the sequence of the inhibitor here. Just to give you a sense of the simplicity of the sequences, they are represented here, so they are 20 or 25 bases long, which is very, very simple if you compare to a standard gene. The typical length of a protein encoding gene would be a kilobase or 1,000 bases or something similar. Right, so this is a very, very, very simple and very simplified.
Okay.
Yes?
I have a question. Here you explain very nicely how you control the nature of the interaction between the components qualitatively. Can you control also the kinetics? And what are the range of time constants that you can achieve with such a system?
The rate constants?
Yeah.
So so you have two parts. So you can control the kinetics. So you have basically you have concentration. So here we discuss just the existence or absence of these compounds, but you have to decide the concentration, which is going to control in part the kinetics. And then the time scales, the typical time scales, are controlled by the enzyme, by the kinetics of the machine, basically. So here the chemistry that we use is DNA processing chemistry. Those are not super fast enzymes. They are fast, but not super fast. And so this is going to more or less control what we get in terms of kinetics. And you will see just on the next slide with the result of this particular experiment. So when we order these DNA strands and we put them in a tube, we indeed observe oscillations. We have a fluorescent reporter that I will not discuss again, but this is time. So you see time is in minutes, and we are over a day somewhere here. And this is the fluorescent level of this system. It's just a test tube where we indeed observe nice oscillatory behaviors. And the time scale, so the period is one on the order of hours.
If you want to vary that period, what is the range you can achieve?
It's easy to make them slower. It's a bit harder to make them much faster. But in principle, it should be possible. We will have an example of where we explore the parameter space of these systems. If I get to it quickly enough, yes. So an advantage of this approach compared maybe to creating those similar systems inside of a cell is that they are relatively chemically simple, as I said, and so it's possible to just write down a kinetic model with detailed kinetic, where we just write all the different elementary or more or less elementary steps. So here you have a cartoon representation of what happened on one of the templates of the previous system. So this guy can bind on the input, on the output, and then by the polymerase it's extended, by the nickase it is cut, and then it can be extended again by the polymerase and so on. So you write all those reactions for all the templates, which is here. And then what you need now is kinetic parameters. So you have two types of reactions. Some involve enzymes, and some are just DNA hybridization reactions. So for simple DNA hybridization reactions, it's possible to directly compute the rates, or to get some good computational estimates of the rates, of all rates. So you don't need to measure them. For the enzymes, you need to measure a bit their characteristic, and this is done by separate experiments. For example, for the exonuclease here, you can give it, you can make a separate experiment, simplified, where you just give it this particular compound, and you look at the decay rate for that compound, and from that you can extract the kinetic parameters that you will later inject in the full model. So the full model looks something like that. It's a bit ugly because you have a number of intermediates, but at least you can put parameters everywhere and solve numerically. And if you do that, you obtain indeed relatively nice predictions of the behavior. In particular, it oscillates, and the period is relatively nicely captured. Yes.
So in that model, how are you implementing the chemical kinetics? They are all kind of low mass action kind of kinetics, or is there something more special into that?
So what we do is, basically we use the law of mass action for the standard DNA hybridization and dehybridization reactions, right? Where we just have a simple first order kinetic, and for the enzyme we use a standard description of enzymatic activity, which is a Michaelis-Menten form. In that case. I also have stuff to say about Michaelis-Menten, but this comes later. Okay, so maybe for the sake of time, I will skip this one, but just the idea is that we can now implement other dynamical behavior, and we select another classic one, which is a bistable system. So here we implement a bistable system by designing this particular circuit here. And then I wanted to talk about the realization of a chemical predator-prey network. So you remember that we need prey replication. So this is this reaction here that we have to implement. And the system that we just discussed are doing that relatively easily, as when you have a double repeat template, then the input comes on the template, and by extension and nicking, you create two copies. So this reaction for going from 1 to 2 is easy to implement in the system. Now we need the predator reaction, which is going again from 1 predator to 2 predator, but you need to consume a prey in the process. So it's a small modification of the system above. Basically, you start from a prey and a predator. They hybridize, so it's again a polymerase extension reaction, where this compound, which is predator-prey, is converted into predator-predator. So it means, in fact, that this guy is just you have a center of symmetry here. So this guy is a palindromic sequence, and so you can implement the reaction. Predator and prey goes to predator, and then we need the chemical sink as usual. So they go to waste using the same enzymatic decay. And indeed, if we look at the individual dynamics, if we look at the individual dynamics of the system, so this is the prey, exponential growth and going to saturation. Predator reaction, again, exponential growth and stopping when there is no more prey, and then decay. And now if we combine everybody together, we indeed observe very nice oscillatory behavior. So here we are looking at both chemical species, so both strands of DNA using different fluorescent reporters. So one is in orange, and the other one is in green. So orange is the prey, and green is the predator. And it happens that because of the reporting system, the orange is reversed. So we observe decrease of fluorescence when there is higher concentration of the system. But if you correct for that and you plot one against the other, you indeed observe the nice limit cycle of the system where you create prey, they get eaten by the predator, there is no more prey, the predator decays, and you go back to the initial state and you start again. And you see, and this answers your question partially. So here you can, for example, you have a control parameter, which is the encoding template. So in ecosystem, it would be the carrying capacity of the system. So we call this template G for grass because it feeds the growth of the prey. And when you change the concentration of this guy, well, you are traveling into parameter space for the system. And so the limit cycle becomes bigger and bigger until they are unstable. And in fact, the system is now converging to a fixed attractor. So it's one thing that is nice with the system is that they are just amorphous, completely amorphous systems. So you can easily compartmentalize them. And you can do that for a number of purposes. First, maybe just to go closer to the biological inspiration. So you can put them into microscopic compartment with the size that is approximately the size of a cell. Or for other possibilities, which I will be describing now. So to do that, we are just using microfluidic systems. So we start from the test tube that was implementing the oscillatory predator prey system. Using a microfluidic, we create an emulsion that you've just seen on the movie, I think. Yes, so here we create an emulsion by just having a flow of this aqueous mixture between two injections of oils, and you create these small droplets that you see there. And then we can collect the droplets, put them on the microscope stage, and observe what is the behavior inside of these droplets. So if we do that for the same predator-prey system that we were just observing in a test tube before, so these are individual droplets, and we see a fluorescent signal, which is proportional to the concentration of one of the two species in this case. And indeed, we see that so when we maintain the function by decreasing the size, which is interesting. And we get thousands or millions or even billions in some cases of individual, very small scale systems that all of them are now small predator-prey molecular ecosystems. So that's nice, but microfluidic allows you to do a bit more. In particular, not to create all those compartments not perfectly equal, but slightly different in terms of where they are in parameter space. And this will allow us to explore the bifurcation diagram of these particular systems, not computationally, but completely experimentally. Right? So what we are going to do here is to modify slightly the microfluidic to create droplets, many droplets, which all of them are the same size, but they don't have exactly the same content. And the content that we will change are those control parameters for the system. So the concentration of the encoding species or the concentration of the activity of the machine. Right? So to do that here, we have split the system into 3 tubes, and assembling these 3 tubes, we reconstitute a full predator-prey ecosystem, except that we do this reconstitution inside of the microfluidic device just before partitioning into droplets, and we change the relative flow rates of this reconstituent. So what happens is that we can create droplets that have more of this one and less of this one, and then the next droplet will have slightly different composition. It's in fact very difficult to track those droplets over time, and we don't want to do that. So what we do is that we also put fluorescent barcodes. So this mixture that contains one particular compound also contains a red fluorescent dye. So by looking at the red fluorescence, we know how much of one particular droplet received of that particular mixture, and we can go back to all the concentration at the beginning. Right? So we do that. We collect many droplets that you can see here on it's a single layer of a droplet on a glass slide. There is a zoom here. You see that they all have different colors because they all have different composition. And then we look at their dynamical behavior over time. And now we can put each droplet back into its parametric position and color it according to the behavior that we have observed. And this is really the creation of a bifurcation diagram experimentally. So this is the process here. We see we are changing the flow rates for the system. This is the place where droplets are created, so it's so fast that you cannot see them, but you can see them afterwards here on this image. And here we just check that we have indeed scanned. So maybe this is important. So this is the representation of the color space that has been scanned by this process. And so indeed we are exploring a three-dimensional cube. So we are varying three concentrations at the same time. And now for each droplet, that you can see the droplet on this image, we can extract 4 different fluorescent signals, one in the green, in the blue, in the red channel, and one which is dynamical behavior in a different channel. So we can put this back into order. So this is again an example. So here we have these droplets, and here you see their behavior. So they have different, as expected, they have different behavior because they have different compositions. It's not very clear that you have oscillations, but if you remove the background, that becomes a bit clearer here. And you can also represent that here on this kind of representation. And here you see that some of these droplets, they indeed display relatively nice oscillations, and some do not. And this makes sense in the end if you put everything back together into this bifurcation diagram, which is three-dimensional in this case. So here all the black points are one observation, one droplet. It has been put in this cube according to the concentration of the 3 control parameters that we have decided for this experiment. And the red points are a set of the set of droplets that shows oscillatory behavior. So it's a bit difficult to see in 3D, but these are slices of the previous thing. And indeed, we extract the fact that oscillations are observed in this area, which is bordered by the expected. We'll not go into the detail, but the expected Hopf bifurcation that you can obtain from bifurcation analysis of this system. But this is an experimental bifurcation diagram of the system showing the different types of behavior that you can obtain from this single system by modifying its parameters, its control parameters. And one interesting thing was that if you write relatively precisely the mathematical equation, you see that there is a very, very small area where you expect a different behavior, which is called hard excitation. It's basically a bistable system where one is steady and the other one is oscillatory. The system can switch from steady to oscillatory in principle. And indeed, we observed a number of relatively weird behavior in these droplets. So these are the time traces. And you see that some drops, they oscillate for some time, and then nothing happens for a relatively long time, a day or so, and then they start oscillating again. And this was observed in many different droplets. So this doesn't seem to be an artifact. And indeed, the droplets that show this behavior, they are located at the back of the Hopf bifurcation here, where where we have predicted that you can observe hard excitation. And so it was possible to create a small stochastic model that replicated this behavior. Okay. Okay, so I want to finish this presentation by trying to extract a few lessons. So I started the introduction saying that we can maybe learn something about biology and biological networks by trying to build them outside of living organisms. So what are these lessons that we can extract? So the first one is about this idea that the network is properly defined or properly explained by a topological representation of the network. Where you have these elements and these links telling you if the links are activatory or inhibitory. And in fact, we discovered that this is a bit confusing. And typically, so people say, okay, yeah, this is a bistable network. I can see that there is two cross-inhibitory behavior, inhibitory interaction, so it should be bistable. But it will never be bistable if the inducers do not form a dimer before acting. So topology is indeed important, but the rates, as you have seen in the previous example of the predator-prey. So the predator-prey network can be perfectly implemented into one droplet, but it will not oscillate just because you are outside of the oscillatory region in parameter space. Or it cannot oscillate because you don't have the proper laws. So your system is maybe not nonlinear enough, and then you will not get a complex behavior with a system that is very close to being linear. Right. And this is the case here. If the inducers do not act as dimers but as monomer, the system is going to be very close to linear, and then it will not implement bistability, for example. Right. Similar for our system here. Again, we've implemented the bistable network, so we could have thought about building it just from A inhibiting B and B inhibiting A. It's not going to work. It didn't work when we tried. If you want to have it to perform as bistable system, because we cannot use this trick of creating dimers in this particular chemistry, so we had to add some positive feedback loops on both sides, and this leads you to having some kind of to obtaining bistability in that particular case. So the topology tells you something about the potential. But if you really want to know what's going on, you need to go a bit into more details. Okay, this is more a lesson that experimentally is creating many problems. But whenever you try to create these systems and you use a machine that has the potential to replicate objects and perform self-replication loops, then you will be attacked by parasites. And parasites also exist in biochemical systems. So some unknown compounds will emerge in your soup and hijack the machinery and the fuel to do their own stuff, which is replicating, and not the system that you want to build. So you have to be careful with that. And also an important point is the role of global couplings. And this is something I will just discuss a bit in more details. So the ideal, so you implement your network as an idealization. So you think that the coupling exists just where you have put them, but in fact it's typically not true. And this comes from the fact that molecular networks, they are typically amorphous. So all species are not physically separated. They exist in the same space. It's just molecular interaction that dictates who is allowed or who will interact with whom. And so the topology is completely controlled by molecular interaction, but molecular interaction is not 100%, right? So when you think you are implementing this network, in fact, you are typically implementing something like this. So you have weak couplings, weak and unintended couplings between more or less everybody. And so this is an example from a simulation that we have done. So we are here simple autocatalytic network. So we expect a very simple behavior, which is exponential growth at the very beginning of the curve and going to a steady state, right? We don't expect any other complex behavior from that very simple network. But let's put it in the same tube as another network, which this one has oscillatory behavior. So the oscillator nicely oscillates as expected. But what we see is that in this system now, this guy is also oscillating, whereas the network is a trivial positive feedback loop. So where does this come from? This is the fact that, in fact, all those nodes, in order to perform the reactions and the interactions that are requested by the network, they call on a common machinery, which are the enzymes, in this case, in that particular, in the chemistry that we implement in our test tube at least. Right? And in a cell, it would be the same polymerase, the same ribosome, the same whatever. And this creates coupling because when this node gets high, so it's replicating very fast, so it's basically using all the polymerase, calling on a lot on the polymerase. There is less polymerase available for this one, so the concentration of this one will decrease. And in the end, you will observe a transfer of oscillatory behavior from one network to the other network just by being in the same chemical element and calling on the same enzymatic and the same catalytic machine. So, so this idea of that you have discrete network structure is a kind of an idealization, both in vitro and in vitro. So these loads, these global loads, they can negatively affect function. So this is exemplified here by computational solutions for again the same network where we switch on and or off the global coupling. So we just mathematically put a 0 in front of the term that describes the fact that they all share the same enzymes. And so you see that without global couplings, this system is a nice oscillator, nicely oscillating. And as soon as you switch on the global coupling, it will affect its function. And in that particular case, it makes the oscillation much less stable. And you move to, you are going to a fixed point. But these global competitions, they can also provide a new function, and we worked a little bit on the idea of implementing the winner-take-all networks. So the winner-take-all is an effect that happens when you have a collection of self-replicating agents, so simple positive feedback loops, and they compete for growth. So they want to replicate, but they all call on the same machine or on the same building blocks in order for their replication. And this is a mechanism that is well known in ecology. It's called competitive exclusion. The idea that within one niche, you cannot have at steady state more than one particular species. So the one that grows faster will, in the end, completely eliminate the other one. And asymptotically, you cannot maintain diversity in this particular situation, in simplified situations, which is, of course, not what is observed in real ecosystems. But that's another point. And so, but the component are very simple. So you have exponential growth and a limited shared resource, and it goes down to discretization. So elimination of all, but the fastest growing species. This is what we have exemplified here. So you start from, so you have this simple, so they share the same resource, they try to grow, and asymptotically, one is in fact decreasing, and the other one is in fact increasing. You have a kind of snowballing effect, where the one that grows a bit faster becomes stronger, and then it takes more resource of the other one, which is going down, so there is more resource for the one that was growing faster, and you snowball back up to a completely discrete situation. So, and that's, of course, from an informational point of view, that's very interesting, because you can get completely digital behaviors without having to implement killing links between all the nodes. And I'm just getting to conclusion when the chair is standing. So I thank you for your attention. Just maybe I'm going to go quickly through. So these artificial molecular networking approach, they provide tools to explore questions related to information processing and networks in molecular implementations. And we now have a range of platforms to play with these things that goes from in vivo platform with synthetic biology to a very simplified platform. Some I presented ones that use enzymes, but some only use DNA and enzyme, but some only use DNA, and they're allowed to explore some question in a very, very simplified settings. And this is something that I've not talked too much about it, but they open way to new applications where you can use molecular systems, not for their physico-chemical characteristics, but as information processing units. Thank you for your attention.
Can we open 5 minutes question before we stop? Yes?
So I would like to thank you for your course. I was trying to think it from the student's point of view. How correct is this and how clear it is, and how correct are you in the most elementary parts? And I think that's where we can discuss improvements, and I'm speaking of significant improvements. You started by saying that the cell is a kind of an amorphous structure, a bag of chemicals. I don't think it's too important to stress the history of what you're saying, that it goes back to 1897, and there's a Nobel Prize for that in 1907. It's related to a controversy between Pasteur and Buchner. Actually, what you're saying is completely ignoring the fact that there are membranes and stuff like that. And the statement you make is, I believe, well, to put it politely, is misleading, but I think it's better to say it's completely erroneous for the students. You are dealing with a system called the cell where, in the first approximation, everything is oriented in space and in time at all scales. And when you say it's amorphous, let's say isotropic, okay, you are speaking of symmetries, whereas we are dominated by asymmetries. I'll give you exact illustrations of what this leads you to.
I think we should let him answer and take out the question. We don't have much time.
I understand. Yeah, I have 6 points.
We should stop here. You can discuss that at coffee break. I think that's
Yeah, we're happy to. Yeah, I made some simplification. That's true. I had a message to get through, and there is one. I mean, for example, it's completely possible to obtain very similar behavior. This is the example of the synthetic biological networks in vitro. So you can obtain similar behavior, for example, in a cell juice where any directions or geometric characteristics have been removed in that case. But we can discuss more.
Any other question? Yes.
Yeah, I'm curious what would happen if you also include like the production of proteins and this kind of stuff, because for now you just talked about DNA circuits or DNA networks, and why did you didn't study the networks with also proteins? It's because DNA ones are simple to study, so you can just
yeah, no, it's a very good question. I mean, some people are creating artificial networks that include protein productions. So they are very close to gene regulatory networks as they happen in cells. And this is perfectly, I mean, that's a perfectly interesting way to do that. In our perspective, we have been trying to simplify the system as much as possible in order to really extract the essence of what can control the dynamic and how you should implement the system with a particular goal. So it's a very synthetic approach that we are applying.
Okay. Yes.
Thanks. I was wondering if you could comment just very quickly on maybe broadly the applications that this technology can be used for.
Applications?
Yeah.
That's fine. So
sorry. So you can have a number of, you can think about a number of applications. In our groups, we are actively exploring two applications. One is in molecular diagnostics, where indeed, I mean, you have inputs that are molecular, and then you want to make a decision based on those inputs. So typically what is done is that you extract the molecular information, so presence of this molecule, and this molecule. You give that to a doctor, and he makes a decision about what's your problem or are you fine, or whatever. But you could think that molecular systems could do this diagnostics task more or less, more independently, in a more autonomous way, right? So they could both detect the biomarker inputs and process this information to make a global decision about what's the state of the system. So that's something we are looking at. And the second application on which we work a lot is implementation of evolutionary dynamics in these systems, because Darwinian evolution is also a kind of a molecular algorithm that seeks to optimize the properties of the system according to some fitness. And so if we have this kind of molecular networks that can look at the characteristic of the system and connect at a molecular level the observed characteristic of the system with its fitness, with its ability to replicate, then you implement Darwinian dynamics, which are not directed toward optimization of standard fitness, but they can be directed toward optimization of externally defined fitness, right? And then you can implement a kind of optimization algorithm that is completely molecular, in principle, autonomously.
Okay, so we have a question from Maria Isabel Moscardo, the remote attendee. So she said, thanks a lot for the very nice talk. I was wondering, how do you distinguish between inhibitors and activators in the ODE formulation?
With the sign.
With the sign?
With the sign, basically, yes. Some positively affect the rate of production, and some will negatively affect the rate of production. So yeah.
Okay, one last. So it's a practical question. You showed that you had in your system, microfluidic system, many micro droplets, and you observe them for like 2 or 3 days. I don't know. Isn't there a possibility for the proteins or the chemicals to diffuse through the membranes and act on other?
Yeah, that's a good question, in fact, so it depends on what chemical you are considering and what is the chemical nature of the boundary between the droplets. In our case, we know that there is basically no diffusion for large charged molecules such as DNA and very large and the protein also, they don't diffuse. But it's possible that some small compound could diffuse, and we know that some, for example, fluorescent dyes or similar hydrophobic compounds, they can go through. So it can be a problem, because then everything will be mixed. It can be also interesting because it can provide a way to implement communication between compartment to compartment.
Is it maybe infected by viruses? So maybe.
Yeah, those parasites that we observe are also DNA molecules, so they don't cross.
Oh.
And so what happens is that when we implement a system in micro-compartmentalized settings, the parasite problem is mostly removed, because emergence of a parasite in one droplet will not contaminate the full system, whereas in a test tube it's going to happen.